Hi everyone, and welcome to today's lecture, which will now focus on protists, plants, and fungi. Now, just a reminder to kind of distinguish this from the previous lecture or some of the other lectures. Protists, plants, and fungi are all examples of eukaryotes, just like we are, just like animal cells are. So if I say human, protists, plants, fungi, what do they all have in common? They're all eukaryotes, which means they all have a nucleus in their cells. Now that's different from the prokaryotes that we talked about in other lectures, such as bacteria and archaea. Okay, and remember, bacteria are things like E. coli, whereas archaea are those prokaryotes that you find in really extreme conditions, like in hot vents. Now with this slide, I want you to make a note for yourself of, how this can come up on exams. I can easily ask you a question saying, which of the following has a nucleus? And I could list out, bacteria, archaea, fungi, all of the above or none of the above. Or I could say it the opposite way, which of the following does not have a nucleus? And I could list out the different examples. So make sure you're comfortable as to what we discussed that are eukaryotes, which ones are prokaryotes, and the examples of those, what it really means. Now, when we mention that protists are eukaryotes, I wanted to give you a definition on this slide because it's kind of interesting how we define protists. Protists are all eukaryotes that are not land plants, fungi, or animals. Now, it's funny because usually you define an organism based on a very particular trait, that all of those particular organisms will share. But there's no single trait that makes a protist a protist. We simply categorize them as being eukaryotes that do not fall into any of those other categories. And I listed out some examples that we're going to see throughout the slides of today's lecture. And I want you to know anytime I mention any of these examples, whether it's in a slide or in a question on an exam, you should know that these are examples of protists, okay? They're not plants, they're not fungi, they're not animals, they're not bacteria or viruses. These are examples of protists. Algae are protists, amoeba, plasmodium, most dinoflagellates, euglena, diatoms, slime molds, and a few others that we'll also encounter. But these are the main names I want you to associate with being protists. Now, in a lot of the things that I teach to my students, I like them to understand why exactly is it important to study that particular topic. And certain topics are easier than others to really label. Why do we study it? Because they're things that you hear all the time. Now, the term protist is not something you encounter in your everyday life. So you may not know, well, why is it so important to study them? Well, one of the big reasons it's really important is that they're medically significant. Okay, we're going to give some examples in the upcoming slides, but there are protists that cause major problematic illnesses. Okay, so it's important to study them to help understand the illnesses that they can cause and then to help try and treat those particular infections. They're also ecologically important. So we're going to go through, some examples of where you see them in food chains and out in the environment. And then the last bullet point here is that protists are important in helping us understand evolution. Okay, because like we said, protists are an example of eukaryotes, which means that there are some connections with the other eukaryotes, which include plants, fungi, and of course animals, which includes ourselves. So in that last bullet point, biologists study protists to help us with those phylogenetic trees that I showed you in other lectures, and with better understanding, ancestry and morphological similarities or differences amongst different organisms. Now we're going to go into a little more detail of each of these bullet points in the upcoming slides. Okay, now the first example of why we study protists is the example of Plasmodium. And I want you to, circle star, highlight the fact that Plasmodium is a protist that causes malaria. Okay, so I'm sure you've all heard of malaria. It's endemic in Africa, meaning that it is always present in high amounts at that location. But you can catch malaria in, technically in any location as well, even though, the real hot spot is Africa. Now, when it comes to malaria, the Plasmodium, as you can see in this diagram here, ultimately causes what's called the lysis of red blood cells, meaning they burst red blood cells. And when the Plasmodium does that, the patient then will experience a variety of symptoms that include things like headaches, fever, really, really bad fatigue. If you kind of think about, people who are anemic, fatigue is a big one. So any of the stuff dealing with your blood cells usually can lead to problematic fatigue as well. Now, when we look at this, I want you to keep in mind that this Plasmodium, which you see these little guys here, okay, are entering. So they're entering through the mosquito saliva, when you get bitten and they infect the person. But I want you to keep in mind that I can, for instance, ask you, which of the following causes malaria? Is it a bacteria? Is it virus? Is it a protist? Is it, none of the above or whatnot? So make sure you're very comfortable in these lessons when, when we give you these different terms, really relate them to the big concept of the lesson. So today we're talking about protists, plants, fungi. Well, what's Plasmodium? It's a protist causing malaria. And what does it end up doing? It ends up causing the lysis, or the bursting of red blood cells, which causes things like headache, fever, fatigue, all this not so great stuff. Another medical example of protists, meaning another problem for you medically that protists can cause, are what we call harmful algal blooms, okay? Algal meaning algae. Algae, things like plankton, dinoflagellates. So in the case of algal blooms, what we actually mean by a bloom, that's when these unicellular species basically have a very rapid population growth, okay? And so you end up with these algae populations reaching such a high density in this aquatic environment that you can actually see in the figures here, you end up with this red tide, okay, this crazy discoloration of this massive waterway because the algae have become so dominant. Their population has just skyrocketed, okay? Now, the problem with this is that ultimately, even though you're not going to go out there and drink that water, you're not going to go eating algae, what you might do is eat seafood that's been in those waters without you knowing it. So for instance, clams and other shellfish may have fed on these red tides without you knowing. And what happens is these algae, or these dinoflagellates, they sometimes produce high levels of toxins. And those toxins can build up in that seafood, in those clams, in those, shellfish. And then you eat them. And now it's amplified because you now get from multiple clams, or you get from that one clam the multiple algae toxins that it took in. And one of the things that this can create a problem with is you can end up with shellfish poisoning that can even be paralytic shellfish poisoning. And paralytic shellfish poisoning, basically, it can range from, let's say, you getting a little bit of tingling in your mouth and your tongue, all the way up to full paralysis of things like your respiratory system. And what happens if your respiratory or cardiovascular system get paralyzed? Boom, you're dead. Okay, so this can be a very big problem. And again, all of this caused by little old protists, which in this case was algae, specifically usually dinoflagellates, you'll hear. Now, in that previous example, we kind of touched upon the idea of clams, shellfish eating algae or protists, and then, bigger predators such as ourselves eating those things. And that then guides us into this slide where we see that protists are also important for their ecological significance as part of aquatic food chains. Now on this slide, you see the difference between a food chain versus a food web, okay? So in a food chain, you'll see how nutrients or feeding relationships exist in a linear fashion. So you're looking at one organism eating another organism, getting eaten by the next organism, whereas a food web shows you all the interconnected relationships of those nutrients in that big population. So it's a food web is basically many food chains put together, and protists are a big part of these food chains and food webs because you see them here as the starting points. So here as phytoplankton, here as dinoflagellates, even diatoms. Okay, so you see them as primary producers on a lot of food chains and food webs. So circle star highlight the term primary producer. Okay, and basically what that means is they will be the species that produce chemical energy, and then they get eaten by other species, other organisms that then gain energy from them. Okay, so primary producers are usually what we call photosynthetic. So they're able to make their own energy, and they kind of serve a major base for food chains and food webs, providing, this primary energy for these systems of nutrient transfer. Now, the main category of primary producer that I want you to know for protists is plankton. Okay, so circle star, highlight the term plankton, and make sure you know that that is an example of a protist. Now, that's one of the things that I can easily do throughout these lessons when it comes to exam time and kind of testing, your knowledge of terminology. I can easily ask you a certain term such as, plankton or algal blooms or plasmodium, and ask you what is that an example of. And any of those will be examples of protists. So now that you know that it's important to study protists, the million-dollar question is, well, how exactly do they study protists? And one of the main ways that they were able to discover, identify, and study protists has been through microscopy. Okay, and at first it was through light microscopy. But then as advancements progressed, we also began to use things like electron microscopy. So as you can see here, we're able to get a really good visual of the overall structure and in some cases the movements of these protists, because a lot of them are microscopic, meaning for a lot of protists, you would have to use a microscope in order to actually be able to see them. Many you can't see with your naked eye. Now, I said for some of them you can also see their movement, and movement we call motility. Okay, so motility is the ability for an organism to move on its own. And the big major protist motility structure that we tend to study is the flagella. Okay, so flagella, and that's all of these extensions that you see coming off of here. Those are little hairs on this long flagella structure. Okay, so this long structure here is the flagella, and some organisms, they'll have one flagellum. Other organisms will have multiple, and what these flagella do is they kind of act like a little whip or a propeller, and push that organism forward. We see this a lot with various bacteria also, not just protists. So a lot of bacteria also have flagella. Even sperm cells have flagella. Okay, so make sure you're comfortable with the answers to these questions. Now, microscopy is not the only way that scientists study protists. A second way that they study protists is through phylogenetic trees, which we've seen before, okay? And when they study them with phylogenetic trees, they're looking at things like synapomorphies, monophyletic groups, paraphyletic groups, all terms that we've covered before that I want you to know the definitions of. So a synapomorphy, remember, is a shared derived trait. So for instance, a synapomorphy that we looked at previously between dogs and humans was having limbs. Okay, so limbs can be an example of a synapomorphy that's a shared derived trait. Now those synapomorphies then distinguish the major monophyletic groups. Okay, so monophyletic group simply means a group that has a common ancestor and all of its descendants. Okay, so a monophyletic group is a common ancestor and all of the descendants. For instance, here is a green monophyletic group, a purple monophyletic group. Okay, so you notice all of the descendants are included in the highlighted area, whereas a paraphyletic group is when it's not all descendants are included in the group. Okay, so some descendants of a single common ancestor, but not all, are included in the group. That makes it monophyletic. So for instance, that makes it paraphyletic. So for instance, this here blue one, the way it is highlighted right now, is a monophyletic group. It has the common ancestor and all of these descendants highlighted. If we suddenly did not highlight diatoms, so you had water molds highlighted and brown algae highlighted, but not diatoms, that would be paraphyletic because you left out some of the descendants. Okay, some of the descendants do not have what you are studying. So monophyletic includes all descendants of that common ancestor. Paraphyletic includes some, but not all. And again, synapomorphy is a shared derived trait. Okay, so for instance, a trait that all of these green plants, these green algae or red algae, all of these guys here in this monophyletic group would share that trait, that would be a synapomorphy. The last technique to study protists that I want to mention is one that is frequently used to identify or discover new lineages of protists, and it is direct sequencing, which is a fancy way of saying that the scientists will sample, the soil or the water that the protist is in, and they'll analyze the exact DNA sequences of samples within that soil or water. And a lot of times it will be called direct sequencing and metagenomics, which we've mentioned in previous lecture. So metagenomics is when you're looking at all of the genes, all of the DNA within a particular population or sample. Okay, so we're not going to go into the details since this is just general bio, but I want you to know that direct sequencing and metagenomics are used to study new lineages of protists, and you can see here the kind of analyses that it includes. So now one of the questions we ask ourselves as scientists is, well, how exactly did protists and other eukaryotic cells end up evolving into those cells that have, all of these internal membranes? Because remember, that's what makes a eukaryote, is that it has all of these internal membranes, structures like the nucleus, like their mitochondria and various organelles throughout them. Now, the main theory used to explain how these eukaryotic cells evolved is called the endosymbiosis theory. Okay, so you have the spelling right here in the figure. And you can break down that word to kind of figure out what this theory actually means. So whenever you see the prefix endo, endo means within, okay? And then symbiosis is when two organisms are living in close connection. They have a very strong relationship. So you have these organisms living within each other with a close connection, and that's because the endosymbiosis theory is actually that cells started out as prokaryotic cells, and then one prokaryote engulfed a second prokaryote. Okay, so you have this big blue prokaryote, it engulfed through, for instance, phagocytosis, a second prokaryote. So you got this, it's engulfing another bacterium. And what do you notice now? Well, you have that prokaryote that had an outer membrane is now inside of the second prokaryote. Okay, so now you have that outer membrane has become an inner membrane within the second organism. And ultimately, this theory of, prokaryote engulfing another prokaryote to lead to internal membranes is used to explain the various, development of these organelles within eukaryotic structures. Now, speaking of engulfing and phagocytosis, that brings us to the idea of how exactly do protists obtain food. And there are three methods that I want you to know for how protists obtain food. There's ingestive feeding, kind of like when you ingest food. There's absorptive feeding, and there's photosynthesis. So for ingestive feeding and absorptive feeding, for both of these, I want you to write heterotroph. Okay, those are both heterotrophic feeding mechanisms. And then for photosynthesis, I want you to write autotroph. Okay, so that's heterotroph versus autotroph. And if you remember, heterotrophs get organic compounds by feeding on other organisms, whereas autotrophs produce their own food. Okay, so heterotrophs will feed on other organisms or other substances, and autotrophs are capable of making their own carbon-based products, basically, as their food source. Now, when we talk about each of these, ingestive feeding, that's basically what you see in the figures here. Ingestive feeding is when a protist uses phagocytosis. So as you can see here, phagocytosis is engulfing, kind of like forming a mouth and ingesting its prey. And sometimes it even uses pseudopodia, which are basically fake feet, fake hands. So you can see here this pseudopodia term, fake feet, fake hands that end up being kind of like a hand and a mouth at the same moment, because they use that pseudopodia to grab their prey and then to pull it into them, so kind of like sucking it down, ingesting the prey to then break it down for nutrients. Sometimes they also use cilia, which we've kind of mentioned before as something that cells can use to move around, but they can also use them to kind of pull food items into their gullet, into their ingestion ability. There's also absorptive feeding, which, as the name suggests, they absorb nutrients from their environment. The examples that I want you to know for absorptive feeding, that would be decomposers and parasite protists. Okay, so if it's a decomposer or parasite, it's absorbing nutrients around it. And then photosynthesis, the third one that we have listed, you've all heard of that. That's when a cell or an organism uses sunlight for energy. And it's basically using carbon dioxide for carbon to then be, building blocks for everything that it's got to produce in terms of biomolecules. And that's why I said it's an autotroph, because if it's using inorganic carbon dioxide for its, carbon to obtain carbon as its source, which it does in photosynthetic organisms, that makes it an autotroph. Okay, so the two examples of heterotrophs here are ingestive feeders and absorptive feeders, whereas photosynthesis, that's an example of protists that are autotrophs. Now another, segue into the next topic. In that previous slide, we mentioned cilia and how many cells or organisms use cilia for movement. And that brings us to the idea of how do protist cells move? And there are three main motility structures that these organisms can have. You've seen some of these motility structures when you look at or study bacteria also. So the three structures I want you to know for protist motility are the flagella, which you see here, and we've mentioned flagella before as basically whipping or propeller-like movement. So there'll be a long structure that looks kind of like a whip, and they will wave it around or propel it around to push themselves forward. And some cells will have, just one flagellum, and others will have multiple, one on each side. Some have flagella all along their perimeter. It varies cell to cell. But flagella is one form of motility. Some other protists instead use cilia, which are these shorter hair-like structures that they wave around to propel themselves or move themselves forward. So whenever you hear hair-like structure, we're usually talking about cilia. And then the third one, which we've mentioned in previous slides already, are pseudopodia. Okay, so a pseudopod or pseudopodia, that's like fake limb, okay, because they look like little feet or little hands extending out. And the cell will basically use those to grab and pull themselves along the surrounding environment. Okay, so when we talk about motility of protists, make sure you know flagella, cilia, and pseudopodia are all examples of structures for motility. Okay, that's enough about protists. Now it's time to move on to plants. Now, when we talk about plants, I want you to remember plants are yet another example of eukaryotes, just like our cells, which means that they do have a nucleus and internal structures, internal membranes. When we talk about plants, there are two main types of plants. So there are green algae and there are terrestrial plants. Okay, so make sure that for two main types of plants, you put green algae and terrestrial. Actually, it's easier if you just write it as land plants. Okay, so there's green algae and land plants are the two types of plants. And you kind of can think about where you would find them. Where do you find algae? You find that in water, right? So green algae you find in aquatic environments, whereas land plants, as the name suggests, you find them in terrestrial environments, meaning in the soil on the land, okay? Now, just like when we went over protists, once you know what the organism is, one of the next questions you'll want to ask yourself is, why is it important that we study them? What value do they have to us? I know it's a kind of a selfish way of thinking, but that's what we do when we pick what we're studying in the sciences. And when it comes to plants, whether they're in the water or whether they're on the land, they're very valuable to us because they're very valuable to ecosystems. And remember, an ecosystem, what we consider an ecosystem is all of the organisms in a particular location along with all of the physical components around it that it's interacting with. So an ecosystem is not just the living organisms, but it's also the atmosphere, the rain, the water around it, the sunlight, and even things like the soil, the dirt around it. And so plants, whether it's green algae or land plants, they're all really important to that ecosystem, to the other living organisms around them, as well as to the ground and the air and the water around them. The reason that is, when we ask ourselves, what exactly do the ecosystems get from plants? There's a few things I want you to write down. First of all, as you probably know, plants produce oxygen, which is very valuable to the ecosystem and ultimately to us as well. I think you all kind of like to breathe oxygen. I know I do. And so production of oxygen is very important, significant part of what plants, whether they're algae or land plants, can do for the ecosystem. They also help build and maintain the soil around them. They also help hold on to water, and interestingly enough, I want you to write down, they also moderate climate. Okay, so they hold water and they moderate climate. What that means, if you think about it, when you are in an environment that has a lot of big plants, aka giant trees, lots of shrubbery trees around you, what do you notice? Those plants, those big leafy plants, they're buffers. They're buffers of the wind, and they're buffers of the sunlight, that heat. It protects, that from reaching whatever is near them or underneath them. Okay, so plants can moderate the climate or help control the climate based on, for instance, buffering wind and buffering heat or sunlight. And of course, as we've kind of mentioned when we talked about some of the other organisms in this course, plants are also primary producers. And remember, that means they're able to convert energy from sunlight into chemical energy that can then be used by the ecosystem around them. And they do that with photosynthesis. So for what do ecosystems get from plants? I want you to write down, plants produce oxygen, they build and maintain soil, they hold on to water, they moderate climate, and they are primary producers. Now, of course, as the selfish little beings that we are, the next question is, well, that's great for the ecosystem, but what do we get from them as humans? Okay, well, there's a lot of things we get from them. First of all, we get all of those benefits that I just mentioned for the ecosystem, because guess what? We live in the ecosystem, right? So we get all of the benefits that we just mentioned from the ecosystem, plus from plants, we get food, we get fuel, we get fiber, and we even get building materials or materials to use for medicine, for the medical field. Okay, so things from plants have actually helped save us many times. And I always like to throw in a figure like this into my microbiology or general bio courses, because I think sometimes people take it for granted that a lot of those heavy-duty prescriptions and medical techniques and all that we get, we've actually gotten from basic biology understanding of even, simple things like plants and foods around us. So you don't always have to, take the heavy dosage pills that you see all around you or that you see marketed in hospitals and all. You can also look to your diet to feel better, to help you with some, common ailments that you might be suffering from. And so, I always recommend looking into nutrition and looking into the biological premises of a lot of the things you eat and drink. It can make a huge difference in how you feel, because a lot of the advancements that we got in terms of the medical field, we actually got from just the things you encounter in everyday life. As scientists, we notice that certain molecules, certain leaves and fruits and vegetables, they have powerful qualities. Okay, so I always recommend looking into some of the dietary changes that you can make in your life. And that helps you to see why it's so important to study plants, whether they're algae, the green algae, or regular land plants. Now, I want to spend a little bit of time on telling you some facts or some important points about land plants specifically, okay? Because remember we said plants we usually group into either green algae or land plants. And land plants, there's some, important things I want to point out. Now, when it comes to land plants, there are 3 categories that I want you to know. There are the non-vascular plants, there are the seedless vascular plants, and there are seed plants. So circle star highlight these 3 groups of plants. Now, when we talk about vascular, so you notice that some of those mention vascular. Vascular tissues on plants are basically just these specialized cells that help conduct water and nutrients across the different parts of the plant. Now, when we talk about the categories of plants, you notice that these first 2, you don't have mention of any seeds, okay? The last one has seeds. For A and B, for non-vascular plants and seedless plants, I want you to put a little star above here and write that these 2 categories rely on spores. So that's S-P-O-R-E-S. They rely on spores to disperse their reproductive cells, whereas seed plants rely on seeds that then get ingested by, things like birds or other animals to help disperse their reproductive cells. Okay? Now, again, we're not going to go into too much detail with any of this stuff. You do that in upper level classes, but I do want you to know the main terminology and the idea of which ones have spores, which ones have seeds that you see on this slide. Now, this slide here, I just threw it in a figure for you to see the evolution of the land plants throughout time. And ultimately, it comes to the point now where angiosperms are what's really abundant. And those are things like you see here, like fruited plants. Now, you don't have to memorize the 5 major intervals, but I want to point out that it's important that now, through the evolutionary times, angiosperms are what's the abundant type of land plant. And so on the next slide, I'm going to give you 3 terms that I want you to know. I want you to know angiosperms, gymnosperms, and the concept of seeds that we just briefly mentioned on the previous slide. So here are the 3 terms that I mentioned that I want you to know and be able to, kind of recognize when I'm describing any of them or knowing the difference between the terminology. The first term is a seed, and the seed of a plant is actually the embryo of that plant with all of the nutrient tissue surrounding it, okay? Basically to help feed that embryo. You also have the terms angiosperm and gymnosperms, and both of those are examples of plants that have seeds. But the difference is that, as you saw in the previous picture, an angiosperm has its seeds encased or basically protected either inside of a fruit or inside of a flower, okay? And then the flower opens up for dispersal after. Now, keep in mind, if I say flowering plants, you then have to know that I'm talking about an angiosperm. The alternative type of seed plant is a gymnosperm. And if I mention a pine cone, okay, cone type plants, they have naked seeds, meaning that the seed is not deeply surrounded and embedded within a flower or a fruit. Instead, if you look at an open pine cone, those seeds are actually exposed. Okay, I know it may from the outside look like it's a protective covering, but it's actually all open and exposed when you look at the ripened seeds. Okay, so if we talk flowering plant or fruit, that's an angiosperm and there is an encased or enclosed seed, whereas gymnosperms would be the pine cone example where it's a naked seed. Okay, it's not surrounded by a fruit or a flower. Now, when we talk about plants living on the land conditions or terrestrial conditions are actually kind of intense. If you think about it, there are periods of dryness, there's a whole lot of sunlight and heat, which can lead to water loss, which you don't want if you're a plant, because as you know, water is very, very important for plants. So plants have had to develop certain structures and produce certain items in order to really thrive and survive in the terrestrial climates or the terrestrial environments. Okay, so the first question that I ask of you on this slide is, what two structures did plants develop to prevent water loss? And the answer is the cuticle and the stomata. Now, stomata is plural of the individual stoma, which you see over here. So when we look at these two structures, the first one is the cuticle. And the cuticle is actually this waxy, watertight seal that covers the various parts of a plant that you see above ground. Okay, so it's this waxy cover, and that helps these plants, survive in dry areas. Okay, it helps them hold on to and retain their water rather than quickly losing water in these types of environment. Now, it was a problem in terms of, the idea that if you cover leaves or plant structures with this wax, well, how do they exchange gases? Because as you know, it's very important for photosynthesis and for the everyday life of a plant to be able to exchange carbon dioxide and oxygen through their plant structures. And so what they also then developed was the stoma or stomata that you see as the second item. And the stomata are basically, they're made up of these openings or these pores that are basically controlled by what we call guard cells. And so at certain times of day or at certain points, the stomata open, at other times they close, and that helps them regulate, controlling and holding on to water during times when water loss might be, more prominent. Okay, so cuticle is the waxy covering of plant structures such as leaves. Stomata are the pores or the openings that open and close to kind of help prevent water loss as well. Now, there's also the idea of intense sunlight constantly hitting the plants. And so in order to properly survive and not get damaged by UV, which as you know, UV can be a big problem for DNA in any organism cells. It can really cause, mutational problems there. So plants developed the production of what's called flavonoids. Okay, and flavonoids are these UV absorbing compounds. So by absorbing the UV, the flavonoids then protect the cells and the structures within the plant from getting damaged by the UV, especially the DNA. Okay, so make sure you know all three of these terms and the main function of them. Now, when we talk about plants, a lot of people focus on flowering plants, so I did want to throw in the main structures and functions of the parts of a flower, and I want you to circle, star, highlight the terms associated with the male parts of the flower, and then the terms associated with the female parts of the flower. Okay, so when you hear anther and filament, please know that these are the structures responsible for producing pollen, and the filament is basically there to hold up the anther. Okay, and those are the male parts of the flower, which make up the stamen. And then the pistil, or the traditionally female part of the flower, that is made up of your structures that you see here. The stigma being the sticky part where the pollen will stick to and be able to make its way through the style and reach the ovary. Okay, and the ovary, just like we have in humans, is a very important reproductive structure. And that'll hold the eggs for fertilization, which ultimately the ovary then becomes the fruit, if that is the fruit type of plant. And you'll see that in the next slide as well. Okay, and that's filled with the little ovules that you see in the diagram. Okay, so make sure you are comfortable with the terminology on this slide. Circle, star, highlight terms like stamen versus pistil, and anther, filament, stigma, style, and ovary. Those are the key terms that I want you to know on this slide. Now, we just mentioned the fruit of a plant, and what we mean by the fruit is basically the structure that's derived from that ovary, that reproductive organ of the flower that you just saw, and that ultimately encloses or surrounds the seed, or if it has multiple seeds, the many different seeds. And the tissues that are derived from that ovary that make up the fruit are usually filled with a lot of nutrients and are brightly colored, which helps attract animals to them. And now this is important because the main purpose of fruit is to actually help disperse those seeds, okay, those future reproductive offspring. Now, the reason why fruit help disperse seeds is, think about it. I know it's a little gross if you think about it too much, but if animals such as birds are attracted to these bright, delicious looking fruit, what are they going to do? They're going to eat them. And then, as the book tells you, everybody poops. So the animal then excretes the remnants of their meal, and that includes the seeds that have gone through their body but have not been digested, because they're nice, indigestible structures usually. And now they've taken them to whatever new location they've flown to, or they've walked, ran, wherever they've gone, they've now put those seeds further away from the original plant. Fruits also help disperse the seeds even when no animal is around, because think about it. An example that the textbook gives is coconuts. The coconut gets blown off the tree by the wind, okay, rolls farther away, or it gets blown off or rolls into the water, and now it's traveling, being carried by the water, and it's a nice protective covering until it then reaches new land, okay, and it ends up helping to move seeds further away from the original plant and increase the chances of spreading those reproductive offspring. Now, the last thing I want to mention with regard to plants is that when you are looking at flowering plants, there are two terms that you tend to come across with these angiosperms. There are the monocots and there are the dicots, and that comes from monocotyledon and dicotyledon, and this is how you spell the word cotyledon. So a monocotyledon has a single cotyledon, and a dicot has two cotyledons. And I know it sounds like a funny word or weird word. A cotyledon is simply a structure that stores nutrients and supplies those nutrients to the developing embryonic plant. So it's very important for that developing embryo. So again, monocot has a single cotyledon, whereas a dicot has two. And we're not going to go into the crazy details about the differences between those organisms, just what it means in terms of the number of cotyledons and how you can tell simply by looking at a flower. So I want you to point out that all of this up here, these figures here, are for monocots. And all of these down here are for dicots. So a monocot flower will have petals in multiples of 3. So either 3 petals, 6 petals, 9 petals, okay? It's a multiple of 3 petals. Whereas if you see a flower that's a multiple of 4 or 5, so if I say a flower has 4 leaves, a flower has 8 leaves or 5 leaves, or 10 leaves, okay, multiples of 4 or 5, that tells you that that plant is a dicot. Okay, so monocot would have multiples of 3 petals, dicot would have multiples of 4 or 5. Okay, make sure you circle, star, highlight that fact. That's an easy exam question that I can ask you. If a flower has 6 petals, does that tell you it's a monocot or a dicot, for instance? Now, finally, we head to the last portion of this lecture, which is on fungi. And I love fungi or fungus talk in terms of microbiology. So if you find anything interesting in terms of fungi, make sure you take our microbiology course where we go into a lot of detail as to things that can happen because of fungi. But in the next few slides, we're just going to give you a quick introduction just as a general bio discussion. Now, even though in microbiology we go over some nasty infections you can get from fungi or some really bad toxins that you can get, the fact is that when we talk about fungi, and if you were to look at all of the fungi in the world, most fungi are mutualists, not parasites. Okay, there are very few fungi that are parasites in comparison to the number of fungi that actually exists. And when we talk about mutualists and parasites, we're talking about those different types of symbiosis, relationships between organisms. So the fact that most fungi are mutualists, it means that most of the time fungi will have a relationship with another organism where both organisms benefit from that close relationship. Whereas parasitism is when one organism benefits while the other organism is harmed, okay? So most of the time, most fungi are mutualist. There's also the third type of symbiosis, which is commensalism, where one organism benefits and one is unchanged. But when we're talking about fungi, I want you to focus on the fact that most are mutualists, meaning most fungi are actually not harmful. They're not pathogenic to you. Okay, there are basically, no more than approximately 300 species of fungi that can actually cause you illness. And that's out of hundreds of thousands of species of fungi. Okay, so please circle, star, highlight that fact, because I tend to ask about, what are most fungi. Now, when we talk about fungi, just like we did with protists and with plants, it's important to ask yourself, well, why study them? And yes, there are those few that can cause illness, so it's important to study them for the medical benefit of treating those few. But on the other side of that idea, the medical benefit of fungi actually comes from what they can give to us rather than what they're doing to humans. Okay, because like I said, most of them are not pathogenic. But from fungi, for instance, we have gotten things like antibiotics. The first antibiotic, penicillin, we got thanks to fungus that contaminated a bacterial plate in a researcher's lab. So the fungus grew on that plate that was supposed to be just Staph aureus bacteria, and that nice fluffy growth ended up showing the scientists that fungi, some of them are capable of killing bacteria, and we were able to extract and use that to our advantage. They also have an economic and ecological impact because, as you can see from these lovely pictures on the screen, fungal infections are a major destruction of crops. Okay, so fungi tend to, cost us a whole lot economically and ecologically with regard to crop contamination. Fungi are also important for the food industry. So if any of you like mushrooms or beer and alcohol, or even things like bread and cheeses, we get these foods based on studying the processes and using fungal specimens. Okay? In fact, fungus is even responsible for us having chocolate. Oh my God, I love fungus. So fungus, without fungi, they wouldn't have been able to ferment cocoa seeds into that delicious, actually edible, palatable treat that you end up eating. Okay? So when I say, why study them? I want you to really write down the fact that there are medical benefits such as antibiotics. There's economic and ecological reasons. And there is the food industry. Okay, but also make note that when we look at fungal infections in terms of medical-based, usage or seeing fungi in the medical field, they're actually much, much rarer, much less common than bacterial or virus infections. Like I said, only about 300 species of fungi get people sick or cause known illnesses out of the hundreds of thousands of predicted species. Now I just want to go through a few examples of terms or types of fungi that you encounter that I want you to be familiar with. The first term that I want you to know is mycorrhizal fungi, okay? And mycorrhizal fungi or fungal network that you see at the bottom of plants, so all up in their roots. Okay, so where do you find mycorrhizal fungi? You find them in the roots of plants. It's a very important symbiotic relationship in which those fungi get things like sugars and nutrients from the plant, and then those fungi help that plant to grow much bigger, much faster, much stronger than they would have without the fungus, because those fungi then provide nutrients and help that plant to absorb things like water from the soil. Okay, so you'll notice that plants that have the mycorrhizal fungi, they grow bigger a lot quicker than plants without them. So again, when you hear fungus, sometimes you think pathogen or yuck, a fungal infection, but having fungus with them is actually very beneficial to some plants depending on what fungus it is. Okay, so mycorrhizal fungi are actually very helpful to plants. Another beneficial type of fungi or fungus that we encounter in our everyday lives sometimes are the saprophytes, or sometimes you hear them called saprophytic fungi. And one of the main places that you find these guys is on dead plant matter. Okay, so a lot of these saprophytes, they make their living by digesting dead plant material, and one of the places that you will actually find them is your very own refrigerator. So when you get that nice fuzzy growth on some of your fruits or veggies, those tend to be saprophytes. And saprophytes are very important, especially in ecosystems. They play very major roles in biogeochemical cycles. So basically, if you think about the cycling of nutrients, of nitrogen, carbon, hydrogen, minerals, all these things, these saprophytes help with that. Okay, they're decomposers, and basically one of the things that they're able to do is they're able to convert the carbon from decomposing organism into carbon dioxide gas. And that carbon dioxide gas then makes its way back into the atmosphere. So that's one of the big things that saprophytes end up doing all around us in the environment without us even realizing it. And then, as you know, that carbon dioxide then plays a major role in things like the life cycles of plants, and which then play a role in our life cycles because we rely on plants. So these fungi are pretty important, even though we don't really take note of them in our everyday lives, or really give them the credit for being awesome that they deserve. Now, when we talk about fungi, there are two terms that I want you to know. Fungi can grow in two possible forms. The first one that you see here on the left, these are yeast cells. And these guys on the right, they're mycelium or mycelia, okay, depending on whether it's singular or plural, okay? Now, you notice the difference between them. Yeast cells are smooth, unicellular little organisms or fungi, whereas mycelium, I want you to underline the capital M. of mycelium, and right next to it, multicellular. So underline the M of mycelium and the M of multicellular, because these fungi, mycelium form, is multicellular, and a lot of times you'll see that they have fungal filaments growing on them, which are known as hyphae. So whenever you see hyphae, those are the long extensions, almost like cobwebs, spider webs, extending out. When you see fluffy fungus, those fluffy extensions are hyphae, and they're very important to the fungi that can produce them, because they allow these fungi to really get into every little crevice of something and really attach to that surface, grab a hold of these surfaces and grow further and further out. Okay, so I want you to know the terminology of yeast, mycelium, and hyphae. You should know that yeast, with that y sound, think unicellular growth form. So unicellular fungus is yeast, whereas multicellular fungal growth is mycelium. And if there are fungal filaments extending outward, those are called hyphae. And that also basically affects the capabilities of the fungus, and ones that are in, cause medical problems, that can also affect the pathogenicity of that fungus. So hyphae are important. The last term that I want you to know with fungi is the term budding, okay? And it's not a term for bros, your bud. Nope. Budding in fungi is a form of asexual reproduction. So where one cell buds off into an offspring cell, and it can even become a budding chain. So a chain of new offspring. And again, that's asexual reproduction. You see this especially in yeast fungi, but you also can see it in some of the filamentous forms of fungi as well. And as you see in the pictures, basically the offspring buds off of the parent cell. Okay? That is it for today. This was just a sampling of general bioinformation on protists, on plants, on fungi. If any of these topics interest you, then I highly recommend you taking our upper level courses, such as ecology, botany, or microbiology, okay? Because we get to go more in depth and really explore some of the cool capabilities of these organisms. Well, that is it for today. Thank you and have a great day.